In this video, I'm going to start by just briefly recapping where we've been and where we're going. And then I'm going to tell you a little bit more about the structure of this particular video, and then we'll dive into this video's topics. So just to look at where we've been and where we're going. So far in this course, we've been very much focused on a lot of things down here at the base of this biological hierarchy, right? So start out looking at molecules, then organelles, then cells. And then as we've been kind of exploring topics about cells, we've examined cell physiology, we've learned about the cell cycle, we learned about how cells divide, learning about the processes of mitosis and meiosis. We've just spent a whole bunch of time talking about Mendelian genetics and linkage, and we've also kind of gone beyond Mendelian inheritance to talk about other types of inheritance. And so now we're going to shift gears a bit to start talking more directly about DNA. So in this video, we're going to talk about the discovery of DNA. Then we're going to explore more about the structure and properties of DNA. And from there, we'll take a look at how DNA actually gets replicated in prokaryotes and eukaryotes. From that point, we're then going to talk about something called the central dogma, and all about how genes get expressed. And then we're going to kind of go a little bit beyond the process of gene expression. So we're going to talk about gene transcription and modification, and then gene translation. So that's going to take us through the next series of lectures from here. But for today's lecture, we're just going to be focused on sort of the discovery and fundamental characteristics of DNA. So the first part's going to deal with how DNA was discovered, and we're going to walk through a series of stories about a group of scientists. So we'll talk about Griffith and his mice, we're going to talk about Hershey and Chase and isotopes and viruses, we're going to talk about Erwin Chargaff's observations, then I'm going to tell you a rather involved story about competition egos in a race for fame. Then we're going to dive into a topic about who gets to write the history of biology, and we're going to talk some about biology's race problem. And then from there, we'll wrap this video up talking about how DNA is replicated. So one last clever experiment from Meselson and Stahl. So the race topic is it can be an emotionally difficult topic, and I'm not going to be asking you any exam questions about this topic, but I do think it's a topic that's extremely important to address in this course, and it's one that historically I don't think has been addressed in an upfront manner as it should be. So to me, it's a really important topic to bring up in this course. And it's something that should be woven into a number of courses in biology, because I think if we don't talk about biology's race problem, then there's really not going to be any opportunity to change it and help the community of scientists move past this sizable problem and issue that's also a larger societal and cultural issue. I should also note that I'm going to talk about this at fairly great length, and part of that is that it's a complex and nuanced topic, and I'm nervous about getting the topic wrong. So I don't expect to get everything right, but I am hoping to at least start more conversation on this topic. And I should also say, I've struggled a little bit with where to talk about this in this course, but I wound up deciding here because of some of the major problems associated with some of the people involved in some of these discoveries. So you'll see that come up very soon. And although I'm going to raise this problem, I'm also going to point out that there are some signs of hope. So from here, let's keep going, and for right now, we'll start out talking about this initial discovery of DNA. And just one other note here. So I do think it's useful to know who's who in this series of stories, but realize that, the who's who is not going to be my major focus here. So our story starts out in 1928 with a biologist named Frederick Griffith. So Griffith was interested in understanding what causes infections. And so he did a series of studies where he started out with some bacteria that were live bacteria that had a capsule around the outside of the cell membrane. And these bacteria were virulent bacteria. So what he meant by virulent bacteria is just if you took some of these bacteria and you injected them into a mouse, that poor mouse died. Okay, so first things first, yes, these bacteria are dangerous. So what Griffith found is now if he took some live bacteria that did not have capsules around the outside of the cell membrane, those non-encapsulated bacteria were not virulent. So if he injected those into the mouse is fine. Okay. So what he did next was he applied heat to those live encapsulated virulent bacteria. So now he has heat-killed virulent bacteria. That should remind you a little bit of Pasteur, right? And remember, these guys are working at a similar time. So if he took those heat-killed virulent bacteria and he injected a mouse with those, that mouse lived. Okay, so heating can deactivate these virulent bacteria. So now it's what he did next that was really interesting, which is he took some of those heat-killed virulent bacteria and he mixed them with some live non-virulent bacteria. And when he mixed those two things together, so neither of those by themselves kills a mouse, but when he mixed those together and he injected a mouse with that mixture, all of a sudden the mouse died. And then if he went back later and he did a little mouse autopsy, so he took a blood sample from that dead mouse, what he was able to recover were some of those live encapsulated virulent bacteria. So he concluded from this series of experiments that somehow these live bacteria were getting some information from the heat-killed bacteria. He didn't really know how they were getting that information, but clearly they were getting some kind of information. That information persisted even after the bacteria were killed. And we now know we now call this process of acquiring that information, of a bacteria acquiring that information from its environment, the process of transformation. But at the time, he did not know what the substance was that these bacteria were picking up. So what is that substance? That was still a burning question. And that was really a larger question among many biologists at the time. How is genetic information exchanged? And it might sound kind of funny to you now, but at the time, the leading contender for genetic information was amino acids. And here's why. So at the time, biologists and biochemists knew there are lots of different kinds of amino acids, right? So we've got 20 different types. And we knew that they occurred in long chains and that they had this primary structure. So that means it's possible to write, very long, complex sentences with amino acids. So, there's a lot of information in that long linear structure in an amino acid. And, if you measured what was actually present in a cell's nucleus, a lot of what's there is protein. So this seemed totally feasible. And, okay, there's this DNA in there also, but there's only 4 types of DNA nucleotides. So at the time, people are like, well, how can you even get information if there's only 4 nucleotides? That's such a limited alphabet. Okay, so while that was going on, at the same time, shortly thereafter, another set of biologists, so Alfred Hershey and Martha Chase, in the 1950s, they're really trying to figure out how viruses worked. And so they focused their efforts on a small virus that infects a prokaryote, the prokaryote E. coli. So very simple E. coli bacteria. So viruses that infect E. coli are called phages. So that's a bacterial virus, basically. So what Hershey and Chase did is, at the time they were working, our knowledge of isotopes had really expanded. So, this is the atomic age. We figured out how to produce different types of isotopes. And so what they're able to do is take advantage of that and also take advantage of a biochemical difference between proteins and DNA. So if you think back to what you know about proteins and DNA, hopefully you will realize that proteins contain sulfur, and meanwhile, DNA contains phosphorus. And there's no phosphorus in proteins, and there's no sulfur in DNA. So what Hershey and Chase were able to do was to create a growth medium for viruses that contained either an isotope of sulfur or an isotope of phosphorus, and grow up a whole bunch of those viruses, and those viruses would then incorporate those isotopes into the new virus. And then what they could do is they could introduce this virus that had incorporated that isotope into a new tube full of bacteria, and then let that virus hang out for long enough to start infecting those new bacteria. And then they could use methods to separate the viruses from the newly infected bacteria, and then check the viruses versus the bacteria to see where their isotopes wound up. And these are radioactive isotopes. They could figure out what was radioactive, what wasn't. So the experiments they conducted are summarized in the picture that I'm showing here. So on the left-hand side, we've got viruses that were labeled with radioactive phosphorus. So when they used the radioactive phosphorus label, what they discovered was the phosphorus was the thing that wound up inside of the infected cells. And then in contrast, when they labeled the viruses with sulfur, so they're labeling the protein in these viruses, when they came back later and they'd separated the bacteria and the virus from each other, they found that radioactive label in the capsule around the virus. And they didn't find any of that label inside of the bacteria. So that allowed them to determine that the substance that viruses are injecting into bacteria is not protein, and instead that substance is DNA. Okay. So again, viruses copy themselves by injecting DNA, not protein. So along with this, Erwin Chargaff made another observation that wound up being an important one in 1952. So here's what he observed. So, again, most people were hyper-focused on proteins at the time. They were discovering a lot of amazing things about proteins. But he went through and evaluated the amounts of different nucleotides that were present in a range of different organisms. So here's a table of that actually from Wikipedia for a wide range of organisms. We've got chickens, we've got grasshoppers, people, corn, octopi, yeast, all kinds of things. And so when Chargaff evaluated the amount of the different nucleotides that were present in this range of organisms, what he found was that the amount of each nucleotide varied, right? So, I mean, if we look at this table, we've got, in the sea urchin, we've got 32% adenine. And then in contrast, in bacteria, we've got 24% adenine. And, we've got 26% guanine there, and we've got 17% in this sea urchin, right? So there's some variation in how much AG, C, and T is present across these organisms. But the ratios of A to T and of G to C were equal to each other. And you can see that in this table too. There's a little bit of variation, but for the most part, again, coming back to the sea urchin, for example, we've got 17% G, 17% T, C, and 32% T, 32% A. So what that means is that, for example, if you add together the amounts of A and G, that was equal to the amount of T and C. Okay, so by that point, people also knew that DNA had a linear sequence to it, but they still didn't quite know how DNA was organized, which was a key piece of information for figuring out how DNA might get copied from one generation to the next. And so, there are a lot of different ideas that got tossed around for how it might be organized, and including an idea that might sound a little strange now, which is that, DNA could have been a 3-stranded molecule. So maybe you could peel a strand off and then replace the one you peeled off. They also knew a couple of other things about DNA's chemistry. So they knew that the purines and the pyrimidines were relatively nonpolar, and that this sugar phosphate backbone had, a very strong negative charge. So you can imagine that nonpolar region clustering together. But they didn't actually know how it was organized. So that then brings us to 3 characters, so Watson, Crick, and Franklin. So James Watson, Francis Crick, and Rosalind Franklin. So another biochemist named Wilkins, mentored Rosalind Franklin in a new technique of the time called X-ray crystallography. And that's an imaging technique that you could apply to a range of different types of molecules. So she applied that to DNA. And basically what X-ray crystallography does is it shows the X-ray scatter pattern that's produced by diffraction through a molecule. So it showed the scatter pattern produced by diffraction through DNA, for example. So she got really good at X-ray crystallography and is able to apply this method to analyzing the structure of DNA. So while Francis Crick and James Watson were puzzling over how DNA might be structured, they wound up having a chance to see some of Rosalind Franklin's X-ray diffraction images, and they noticed something interesting, which is that those diffraction images were very symmetrical. There weren't any lumps or bulges. So then, that got them to wondering, well, how could that be factored in? So they had that idea in mind, and they also knew about Chargaff's findings, and they were also in a very fierce race to be the discoverers of how DNA was organized. And they were racing against another biochemist named Linus Pauling. And Linus Pauling was actually already famous for starting to deduce protein secondary structures, right? So the alpha helices and beta pleated sheets. Okay, so they're in this hot contest to figure out how DNA is structured. And eventually they put these puzzle pieces together to figure out that the way that DNA is organized is that purines pair with pyrimidines and that they hydrogen bond. So their DNA comes in pairs, and that the DNA molecule forms a spiral double helix, so helping to explain the symmetrical diffraction shapes they saw. They also discovered that the two strands of DNA run in opposite directions from each other. So they found that DNA strands run anti-parallel to each other. And once they had figured these things out, it then meant it was possible to start imagining how DNA might work. And there's a historical note here. So Rosalind Franklin died of ovarian cancer at age 37, most likely because she was exposed to a tremendous amount of X-rays, which we now know damage DNA. And during her lifetime, she was not recognized for her pivotal role in this DNA discovery. Since her death, people have worked to recognize her for this crucial role. But, very much like Mendel, no recognition while she was doing the work. And unfortunately, that's often the reality for many people that have made important biological discoveries. And meanwhile, James Watson is still with us, but some things have come to light. So this is an article from 2019 about James Watson and about a documentary that's about him and some opinions that he's expressed about race. And I'll provide a link to this article if you're interested in reading about it. But the bottom line is that Watson holds some incorrect ideas about race, and they're very harmful ideas. And because he's a fairly famous biologist, it's a big problem that he holds these ideas and that he's publicly expressed them. He has suffered some consequences from his decisions on that front, but again, it remains very problematic that he's a well-known figure in biology because he also holds these problematic opinions. And he kind of raises the point or the question of, within a subject or within a scientific community or any community, when there's a problematic individual in that community, what do you do about that? I don't think we really have good answers to that question. We could try to erase Watson from this history, but that would be kind of disingenuous because he did play a role in this discovery about DNA. But my hope is that we can separate that discovery from, the person in this case. So, yes, a person can make a really impactful discovery, but that person can still be someone who holds incorrect opinions and someone who misuses their fame and authority. Relative to, what's really just and right in society. So, we have a choice now about how we talk about these historical figures in any subject area. And so I think over time, what we really have to do is think about more nuanced ways to talk about people who have made important contributions, but who also hold perspectives that are just plain wrong. And part of why I want to talk about this is because I think biology still has a really large race problem. And this is something that can't change unless we talk about it directly. And here's a kind of a contemporary view on this problem. The biggest thing is that race is not a biological reality. Race is actually a social construct. The biological reality is that there is amazing and wonderful genetic diversity within and among groups of humans. And, that doesn't really map onto race at all. So race is a social construct. But we need to talk a little bit more about what I mean here. So I want to be really clear here to say that making this statement does not mean erasing the concept of race, because it's a social construct. So it's something that societies have created and that they use to categorize different people. And primarily, it's been white people that have been using this to categorize and marginalize other groups of people. So, we're very much a visual species. We do very much rely on visual appearance when we're judging people, in multiple different ways. And so, we use visual appearance to categorize people, and that changes how we behave towards different groups of people. So, but that's, if if I describe myself as a white person, that doesn't actually really have a whole lot to do with my genetics. It has, yes, there's going to be some genetic component to pigmentation in my skin, but there, if we look at, the genetic association with any other aspect of my phenotype, that varies tremendously across everybody, and it has no connection whatsoever to the genetic component that determines the color of my skin. So, socially, groups of people have really built a lot around the idea of race, and have attached a lot of implications to having different skin pigmentation, when the biological reality is that yes, there's variation in human skin pigmentation, but there's also variation in a lot of other characteristics. And this is a point where we can think back actually to the example I've given for sickle cell anemia, where it might seem like one genotype is better than another genotype, but you can't really separate that from the environment or the context where you're thinking about that genotype. Because it turns out that one genotype that might be better in one environment is not so great in another environment. And that's an idea that holds true for human populations, and it holds true for other organisms also. So again, just to reiterate here, the way that human societies have defined race is very much a social construct, and it doesn't have a basis in our understanding of the biology of being a human being. So here is an article from 2009 that is a news article about a survey of human genetic diversity. And we're going to talk more specifically about human genetic diversity a bit further on in the course, but I want to start talking about it here. So this Human Genome Diversity Project involved surveying genetic diversity of over 50 geographic groups from across the globe. So it included 51 different populations. And I'll also provide a link to this news article for you. And here are the takeaways from that study. The most important one, I think, is that groups across the world are far more similar to each other than different. So we are all part of the same human family. Along with that, it's very well established now that the majority of people have genetic ancestry that traces back to more than one continent. So the majority of people have ancestry that is diverse, and really what that means is that as a species, Humans are very migratory and mobile. We've moved around a lot. We've, groups of people have mixed with each other a lot over time. Along with that, from this project, the scientists that conducted this project did establish that populations lost genetic variation as they migrated further away from Africa. And it is still possible to trace an individual's geographic heritage through their DNA. But this did not provide support for concepts of race. And again, this is something that is still a very big challenge within biology. So here's an article that just came out mid-September of this year, that's a letter in one of our scientific journals about the use of race in science, and specifically how race is used by the National Institutes of Health. Because people have identified that there are major problems to how race is used. And to give you an example of this, you may have heard in news stories about a lot of differences between different populations of people in terms of susceptibility to COVID-19. And it's fairly easy to make a leap from those differences to thinking that maybe there's some kind of genetic link to susceptibility to COVID-19. And actually, very recently, there have been a couple of papers published that have shown that there is a genetic component to susceptibility to COVID-19. However, the genetic components that have been identified have diddly squat to do with race. Absolutely nothing to do with race. So there's genetic variation in human populations across the globe that affects susceptibility to COVID-19, but no connection to race. So what's actually going on with why there are subpopulations within the US where there's much higher rates of COVID-19? Well, it doesn't have to do with any aspect of the biology of people in those populations per se. Instead, what it has to do is with everything that has to do with racism that has put people in those populations in more vulnerable situations, that has then caused them more likely to get COVID-19 and to suffer some of the worst consequences of COVID-19. So it's not that people of specific races are more susceptible to COVID directly because of something having to do with their human biology. Instead, it has to do with their social and economic circumstances, in a lot of cases which have been caused by racism. So again, I'll provide a link to this article as well. So again, I don't think there's a simple answer to what we do with, problematic scientific figures. I think an important first step is raising the issue that there are scientific figures that are hugely problematic, and that it's inappropriate to put those figures on a pedestal and pretend that they aren't problematic. Along with that, we have a lot of control over how we decide to tell stories about the past. And so I think it's also important to think about if there are ways that we can rewrite those stories that help provide a more realistic contemporary understanding of who those people were and the things they discovered and the context that they were working in. So we don't need to sugarcoat those people. We don't need to put them up on a pedestal. We can recognize that they made important contributions that still impact our thinking today. And we shouldn't try to erase them either, because we're always going to have problematic figures. But I think if we do all of that, that, or I hope that by doing all of that, we can better come to grips with, some of these issues that are at the intersection of science and society. So that our science is not supporting problematic political and cultural practices. Okay, so now I want to come to the last topic here. After discovering how DNA is structured, the next question that came up for biologists was, well, now how is DNA actually replicated? And there are actually three different possibilities for this. So one possibility is referred to as conservative replication. So that's the idea that if here's your parent DNA, that these two strands both get copied. And so at the end of the day, in your next generation, you would have the original parent strand and then one brand new strand. And then the generation past that, you have a single parent strand and then three new strands. So that's conservative replication. A second possibility is that DNA could be replicated in a semi-conservative fashion. So that's the idea. You take these two parent strands, and then in the next generation, each piece of DNA consists of one parent strand and one new strand. And then by the time you get to the third generation, then you have two hybrid strands, and then the two new strands also wind up forming as well. There's a third possibility called dispersive replication. It's the idea that you go from a single parent molecule to two kind of patchwork molecules, and then that kind of patchwork process persists into the next generation. Okay, so three possibilities. So how did biologists get to the bottom of this? So the scientists that sorted this out also used isotopes. And the two biologists were Meselson and Stahl. And so they used a combination of heavy and light isotopes of nitrogen to figure this problem out. And so in the little cartoon that I just showed you, we can think about the black colored strands as being strands that contain heavy nitrogen, and then light colored strands being DNA that has light nitrogen. And the idea that they used was they'd take a bunch of cells and they would transfer those cells from a broth that contained the heavy isotope of nitrogen to the light isotope of nitrogen. And then they'd allow enough time for those cells to go through a single round of replication, and then they would examine the results. And the way they'd examine the results is they would break apart the cells and they would put them into a centrifuge tube. And if you put a substance into a centrifuge tube, you spin it at high velocity, so the force of gravity pulls down towards the bottom of the tube. So you have increasing gravity down here. So things that are heavier are going to wind up closer to the bottom of the tube, and things that are lighter will wind up closer to the top of the tube, or less dense. So in this little illustration, this tube is just turned on its side. So if you took a tube that had cells in it that had just been growing in the lighter nitrogen isotope, you would get a band that is closer to the top. And then if you grew cells in a culture that just had the heavier isotope of nitrogen, nitrogen-15, then you get a band that's closer to the bottom. So if you take this cells that have been growing in the heavier isotope and transfer them to the lighter isotope, and you know how long it takes for replication to happen, then you spin again and you can check out the results. So here is what they saw. Or I should say, here's what you would expect to get if replication is conservative. So this picture on the left-hand side, right? If you did this, then you'd expect in your F1 generation that you would get a mixture of the heavy band and the light band, right? However, what they found was the following. After one generation, they only got a band in the middle, intermediate. So this is consistent with either the semi-conservative replication or with dispersive replication, right? So as with a lot of Mendel's experiments, what we really need here to get to the bottom of the matter is to also look at the next generation past that. So if you look up at these pictures in the corner, what would you expect to see if replication was semi-conservative versus if it were dispersive? So hopefully you can see that if it were semi-conservative, you should see two bands again. You should see a medium band and a light band. Whereas if DNA is replicated in a dispersive fashion, you should still see just that intermediate band. And here is what they found in that F2 generations. They found two bands. So what that means is that's only consistent with semi-conservative replication. So DNA is replicated in a semi-conservative fashion. Okay, so that's a bunch of historic stuff. If we fast forward to now, interestingly, even when DNA is unwound, it remains very, very organized. So here is a modern staining method using fluorescent dyes. And each of these colors, as you can see for the chromosomes down here, each of these colors represents a different chromosome. So even when chromosomes are decondensed, they're still organized, and so the nucleus is very much organized. The density within the nucleus can vary, and so within the nucleus there are some regions that are more densely packed. Those regions are referred to as heterochromatin, and then there are other regions that are more loosely packed. And the heterochromatin regions are regions of DNA that are generally not getting expressed, while euchromatin is a lot looser and is getting expressed. So again, over the next series of lecture videos, we're going to first talk about how DNA gets replicated, and then we'll talk about how DNA gets expressed.